PTMs. You all remember him from his talk on Monday on proteomics. So let's welcome Dan.
So this talk is about computational PTM proteomics. So I guess there's a lot of people in the room that have done some standard proteomics, some full proteome expression, and this PTM stuff, it's just a whole nother level of difficult. From the beginning to the end, everything is just more complicated. So let's have a look at the overview of what I'm going to talk about basically up to this point, and then Stefka is going to take over in the tutorial and talk about everything that you're going to do downstream of the max quant output with Perseus. So we begin with enrichment. Then we have to identify our post-translationally modified peptides. We have to localize them. We want to know exactly where that PTM is on the peptide. We also want to calculate occupancies. Those are biologically meaningful output from PTM-enriched peptides. And as I said, Stefka is going to talk about the quantification analysis in her tutorial. So enrichment strategies then. So I've listed here the different post-translational modifications, beginning with phosphorserine threonine. Then I have the enrichment type, and then a manuscript that you could consider going to get some advice on how to start. So I just want to point out this one, the Humphrey et al. In Nature Protocols. So one of the authors of that paper, Özge Karayel, she's actually here at the summer school, and maybe you can get some practical advice from her on how to do serine threonine phosphorylation enrichment. Another interesting recent development is phosphorylation of histidine. Now, one has to be very careful with this because it's acid labile, and this is described in a recent Nature Methods paper from Albert Heck and Simona Lemire. So you can see that these two use infinity, whereas affinity based on ionic or some kind of chelation interaction as with the titanium dioxide. But a lot of the PTMs can only really be enriched with antibodies. So there are antibodies that are commercially available that you have to purchase. Those antibodies take really a long time and a lot of effort to develop. So you can't really go and just make your own. I want to point out this paper from Akimov with respect to ubiquitination. So they just developed a new antibody for enrichment of ubiquitinated peptides. They actually get a depth of 63,000 ubiquitinated sites, which, for those of you who have done ubiquitination PTM, it's unbelievable. There are, as I think has already been mentioned by Josh Kuhn, for glycosylation, there are specific affinity ligands available that are naturally occurring. And we're also starting to see things like click chemistry being used to get a derivative of the post-translation modification. And that's particularly exciting for the future. So I've just highlighted this paper from Nature Communications. So that's if you know the post-translational modification that you're interested in. From Pavel's talk, we already know that you may have some kind of unknown modification. And there I would recommend to IP the protein to get some kind of purity, and then do a dependent peptide search. So, are there any chances in the future to not have to do these enrichment steps? Right? I mean, this would be like the ultimate goal of PTM proteomics is just to get so deep with our full proteome that we get all of these PTM peptides in there. And I want to highlight this paper from Jesper Olsen's group, where they they basically did that with with the current technology that we have. They went as deep as it's possible to go in a realistic time frame. So I think it was only 24 hours of measurement time, and in that they actually detected 10,000 phosphorylation sites and 7,000 N-terminal acetylation sites. So you can see that as the technology develops, we are going to get closer and closer to achieving PTMs without enrichment. Okay. So with PTMs, we're obviously going to get different spectra relative to the unmodified counterpart. So first of all, you're going to detect a mass difference in the MS1 feature, and this is also probably going to be accompanied by a fairly large retention time shift. And when you sequence the peptides, not all of the peaks, but some of the peaks are going to be shifted by the presence of this modification. And Pavel had a really nice talk about the use of this in the dependent peptide search to use this shift to identify the modification and what peptide it might be on. Okay, so in order to get these fragments, to get these information-rich spectra, there are different fragmentation methods available. So there are essentially 3 common methods: collision-induced dissociation, higher energy collision-induced dissociation, or beam-type CID, and electron transfer dissociation, which we heard about from Josh Kuhn as well. So essentially, what we want is to fragment our peptides, and ideally this would occur along the backbone. We don't really want to just cleave off the side chains. That wouldn't be very informative for us. And fortunately, all of these different methods do essentially cleave the backbone. So CID and HCD give rise to B and Y ions, where they cleave the amide bond. So the B ions have the charge on the N-terminal, and the Y ions have the charge on the C-terminal fragment. And ETD gives rise to C and Z ions, where you break the NC alpha bond. There are some other interesting things about fragmentation. So it can occur where you get B and Y type cleavage together with A and X type cleavage, which gives rise to this ammonium ions. And the phosphotyrosine ammonium ion is particularly informative, because essentially when you see this peak, or a mass to charge ratio of this size occurring in your spectra, you know that there was a phosphotyrosine in that peptide that you sequenced. Okay, so let's have a brief look at the 3 different methods, or a deeper look at the 3 different methods. So classical CID is done in the ion trap. And this is done with a resonant radio frequency. So you try to excite the peptide so that it collides with a collision gas. In there, there's always an inert gas. And this fragmentation produces mostly, as I said, sequence-informative B and Y ions. The problem with CID, because it's done in the ion trap, is that there's this 1/3 rule where you lose these low M over Z fragments. Essentially, they get so excited that they just crash into the wall. And also, because this phosphotyrosine ammonium ion that I mentioned has a low molecular weight, typically you would never see that in a CID spectrum. The other big problem is that you also cleave labile bonds first. So you imagine we take our peptide that's phosphorylated on serine, and you excite it, and the most common fragmentation to occur is that you just lose the phosphate group. So that's what you can see here, right? So you see this is a peptide that's been fragmented by CID, and it has a mass that corresponds to the loss of phosphate plus water. That can itself be relatively useful, because you can see that there was a phosphoserine on it in the first place, but now we don't know the sequence of the peptide. So there are a couple of strategies to overcome that. One of which is an MS3 method, which is done in the ion trap again. So what you do with this MS3 approach is to fragment the peptide to observe this neutral loss peak and fragment that neutral loss peak again. Right? So you read out the MS2 spectra, you see the neutral loss peak, and then you fragment again, and then now you see the peptide sequence. So that's what this MS3 spectra is. Or, which is kind of a nice strategy, is to do both fragmentations in one. So since you expect that this neutral loss is going to occur, you fragment once at the master charge of the intact peptide, and then again at the mass of the neutral loss peak that is expected, and then you measure them both together, and that's called a multi-stage activation. And this is actually still in use. So as I said, even with this taken into account, with CID we still have this low M/Z loss. So one of the things that is also possible is to fragment the peptides in a slightly different way, again using collision-induced dissociation, but this time with higher energy. And so this is typically performed for instruments like QTOFs, but essentially what you do is, instead of exciting the peptide with this resonant radio frequency, you just inject it into a part of the instrument that's filled with gas, and that can be done very quickly. So that's illustrated here. So we have our ions, and they're in the C-trap, and then we lower the potential, and then they go quickly into this octopole collision cell, and then they fragment very, very quickly. And essentially, in using this, you don't allow the peptides time to fragment at the most labile bond, but they fragment all over the backbone, right? So with these HCD spectra, you get the informative B and Y ions, and together with sometimes neutral loss. So this was done. This was a paper from Jesper Olsen in 2007. Initially, this so it's called initially called higher energy C-trap dissociation, because what they did, they actually injected the ions not into a dedicated collision cell, but they injected it into the C-trap, which is also filled with gas. Just that's where the name comes from. And now the thing is, instead of reading out in the ion trap, you can read these fragments out in the orbitrap, which can detect low M/Z range. So the last fragmentation method, which has already been discussed today, was electron transfer dissociation, and this is fundamentally different from CID or HCD. So you actually transfer an electron to the peptide from an anion source, which subsequently undergoes free radical-driven cleavage, giving rise to these C and Z ions, which cleave at the NC alpha bond. The beautiful thing about this is that it's fully sequence independent. So whether or not we have certain side chains or modifications, it typically always gives rise to these nice C and Z peaks that occur along the sequence of the peptide. So this is really useful, for example, with peptides where you want to do de novo sequencing. The disadvantage is that it doesn't work well for low charge density precursors. So if you have a typical tryptic peptide, which is charge state 2, it will not fall apart. So it will break, but it will undergo ET no D that Josh Kuhn described this morning. So there are an increasing number of methods to overcome that limitation. So we heard about photoactivation, but there's also the possibility to do HCD after ETD, and then you get a mixture of these C and Z ions and B and Y ions. But So just to summarize then, with CID we get typically fragmentation of the labile PTM. With ETD we get very nice peaks along the peptide length, as long as we have a high enough charge density. Otherwise, we're going to need to use something else. And with HCD, depending on the stability of the PTM, we can get either a dominance of peptides which still contain or fragments which still contain the PTM or not. So, I mean, in general, most of us, including myself, we're not going to spend a lot of time developing these methods and testing all of the different fragmentations and all of the different collision energies which it's possible to use, which is why we go to the literature and we look for the person in the field who's really developing this, who's the expert, right? So, I mean, if you want to learn about ETD, you look at Josh Kuhn's papers and you see what he's doing. Oh, and one brilliant example of the use of ECD is Ivan Matic with the ADP ribosylation. It made a huge difference for his work. And for phosphorylation, I would advise you to look at the work of Jesper Olsen, who just published this really nice paper comparing lots of different strategies for fragmentation of TMT phosphopeptides using the fusion Lumos. Okay, so what do I have to think about in MaxQuant when I'm searching for these post-translational modifications? It's really unbelievably simple. In the group-specific parameters, we go to the modifications. We look at this box of variable modifications, and those that we want to search for, we just click them over to the right. Is it really that simple? Yeah, it is. Okay, what about all of that CID, HCD, ETD stuff that you just told me about, Dan? Well, where do I put that in? Well, you don't need to worry about that either. It's in the raw file itself. In the raw file, it tells you, it tells MaxQuant what fragmentation was used. So you can just go to the beach. Okay, what about those diagnostic peaks and those neutral losses? What about those? How do I take care of that? Well, for all of the modifications that are already configured in MaxQuant, it's already in there. You don't need to think about it, right? So it's done in the background. There's no user input required. But if you have a new modification, you would go to the configuration tab in MaxQuant, and then I'm just clicking on the phosphor STY here as an example, but then you can see that the diagnostic peak, you would just press plus, and you would then enter the neutral mass and the composition, and it would, you would enter the composition. It would derive this for you, and then if you have neutral losses, you would just put them into here, and I just put these in. This is from Ivan Matic's paper, the neutral losses that they put in that can occur with ADP ribosylation. So you would just enter the fragments that would be expected, and it will search for them. So yeah, exactly. Where are they searched for? We've already had 2 nice talks about the Andromeda search engine, so I'm not going to go into heavy detail about how that works. Suffice to say that there is a few extra things that the search engine now looks for when you have modifications. So obviously it's your PTM modified peptide is going to be in the search engine, in the search engine database. But with, as I told you, with HCD, it may or may not undergo neutral loss. Actually, it's searched for twice. So the score is calculated once with and without those loss peaks. And it will also look for these diagnostic peaks like this phosphotyrosine ammoniamine. Okay, there are a few other parameters in MaxQuant related to post-translational modifications. So if we look at under global parameters and identification, you can see that we have this minimum score for modified peptides and minimum delta score for modified peptides. This is the lowest Andromeda score that we are willing to accept for having identified a modified peptide. So the default value is 40. It's actually like a fairly low value for an Andromeda score if you're familiar with them, and a minimum delta score for the modified was 6. This is saying what's the difference in Andromeda score that we need to see between our PTM modified spectrum relative to the next sequence distinct spectrum. And the default value there is 6. Those empirical values were derived from this library of more than 100,000 synthetic peptides. This was this big community effort that also, by the way, looked at different fragmentation methods and how good they were and how well they were identified. But that's where those values come from. Okay, so now that we've identified our post-translationally modified peptides, we want to localize them. So it's best explained using this example. So in this example, we have a peptide which contains three potentially phosphorylatable residues, a tyrosine, a threonine, and a serine, and we know that it's modified on 2 of these sites. Okay, so how do we know whether it's modified on these two, or these two? Apologies for those watching on YouTube. So we can get the probability back of having seen exactly this spectra by just exponentiating the Andromeda score. And now what we do is we say the likelihood of this tyrosine on the N-terminus having been phosphorylated is just the probability of having seen this spectra and this spectra divided by the probability of all of them. And so on for the other 2 sites. And when you look for this localization score in MaxQuant, in the output, you will see that almost, or a vast majority of the scores are very close to 1. But we can take a cutoff of also of 0.75 typically to say whether or not we have identified it reasonably well. Okay, and that's in this paper here in Cell Reports from Kirti Sharma. Okay, so one of the other things that we will want to obtain from PTM data is stoichiometry. And this is important for biological interpretation of that post-translational modification. You can imagine there's going to be a big difference between something which is modified on 1% of the protein or 100% of the protein, right? That this is important not for anything but biology. So this can be derived with specific methods, and there's a very interesting paper from Chudari's group in Copenhagen, where they did serial dilution SILAC. So in this case, they were actually looking at acetylation, and acetylation for a lot of cases is known to be very, very low stoichiometric. Now it's known anyway. So what they did, they actually spiked in a 100% chemically acetylated set of peptides and looked at the SILAC ratios compared to the endogenously acetylated peptides. And you get this dilution ratio, and they can now see exactly what the stoichiometry must be that way. But this is kind of complicated, and you're not going to be able to do this for a lot of different modifications. So it can be derived from your existing proteomics, PTM omics data. So in order for you to get these stoichiometry or occupancies, we're going to need to have certain information. So not only do we need to have measured our phospho or other PTM, but we also need to have measured the full proteome. So this is the experimental design that you would have to input into MaxQuant in order to get these occupancies back out. So here I have 3 replicates of a proteome, 3 replicates of a phosphoproteome, and they're in different parameter groups. They're in different parameter groups because I only want to search for the modification when I've actually measured the enriched set of peptides. Okay, now they're also labeled according to their experiment. So we want to tell MaxQuant that this is the proteome file and this is the corresponding phosphoproteome file, so that they get combined and so that that's how you obtain these occupancies. I've also numbered the fractions here. So these have all been numbered fraction 1 and these have all been numbered fraction 10. And the reason for that is that only adjacent fractions are matched one with the other. So in this case, all of these will be matched to each other, and all of these will be matched to each other, but not between those two. And lastly, this is a new element in MaxQuant for those that have been using previous versions. Is now this column for PTM. And you'll see that for these it's false, and for the phospho it's true. And this is actually quite important, I would say, because what this means, what this does, is it excludes, especially important for label-free, it excludes any peptide intensity that was identified from that enriched sample. So, for example, if you're using IMAC to enrich for phosphopeptides, IMAC can also enrich for peptides that contain glutamate. And we don't want those enriched peptides, which are not modified, to contribute to the protein quantification of their respective protein. So just one step further is that normally when you want to get these so the reason we measure the proteome is that we need to identify the unmodified counterpart peptide in order to get these occupancies. And to do that, you will need to go deep into the proteome. So single-shot measurements are unlikely to identify all of these unmodified counterparts. So in order to go deep, we would have to fractionate our full proteome. And I've just illustrated how one would set up MaxQuant in order to get the correct strategy and get the right information out at the end. So these slides are going to be available, so you can use this as a template when you have your own data. So how is stoichiometry actually calculated, or like where is it? Where is it? How do we get it? Right. So we have so for SILAC data. We would have 3 different ratios, and we would need all of them. So we would get a ratio for the phosphopeptide, a ratio for the non-phosphopeptide, and also an overall protein ratio. So you can see here that the heavy channel is more intense than the light channel. So we have more phosphopeptide here, and here the heavy channel is lower for this counterpart non-phosphopeptide. So by this we can kind of infer that if all of this was initially unmodified, that this has been modified to a greater extent, and that's why we don't see as much of the non-modified anymore. Okay, so this is the calculation over here. It's described in this paper in greater depth, and you can go ahead and read the paper and convince yourself that the calculation is correct. All you need to know from me is that this is done in the background in MaxQuant. So you don't need to do anything to obtain these occupancies. If the information is in there, if you have these 3 ratios, it will be calculated. There's also a label-free version of this, which works in a similar way, but there's also the addition of this error estimate, because the calculation or the formula that's used for this it can give rise to spurious occupancies, because this can become a near 0/0 calculation, and then the occupancy is just way off. So MaxQuant actually picks. So the difference between label-free and SILAC is that in SILAC we know which ratio we have to pick, right? The one that it was seen with in the MS1 spectra. But for label-free, you can actually take any other peptide for comparison. Or not any other peptide, but any other sample where that peptide was identified for comparison. Okay, so just some use of these occupancies. So I would, first of all, I wouldn't perform your statistics using these occupancy calculations. Stay with your intensities or your ratios. But this is just a histogram of the measured or the calculated occupancy relative to the frequency at which it occurs. And you can see that in the control sample, not so many peptides are found at very high occupancy. And that's really not the case for mitosis, where we see a very large number of our phosphopeptides, in this case, being found at very high stoichiometry. And that's a really interesting biological phenomenon. And you can see here that in EGF-treated cells, that the histogram looks generally the same. But if we now take a subset and just look at the occupancy of the phosphotyrosine peptides from within that, we can see that these are strongly found at higher occupancy. Right, so this is kind of what we expected, right? So that EGF, which signals through phosphotyrosine, we expect to see changes, but I don't know how many of us would have expected to see such high occupancies on all of the phosphotyrosine sites when we stimulate with EGF. And okay, so is there anything else that I need to think about in MaxQuant when I'm doing post-translational modification analysis? So there's a few other things in there. They're all set by default, so you don't need to touch it just to know about it. So under the global parameters in label-free quantification, we have this thing called advanced site intensities, and all that is, it chooses the best combination of peptides for consistent quantification across as many samples as possible, accounting for differences in charge state and also accounting for miscleavages. And why have we done that? Or like why do we even need to do that, right? Because it can be that you will identify one peptide or the same peptide in two different runs. In one run, it will be charge state 3, and in a different run, it will be charge state 2. And because the physical-chemical properties of peptides make it not possible to directly compare the two, so we can't compare them. So what MaxQuant will do is find the optimum charge states and miscleavage states in order to compare the greatest number of samples. So that's turned on by default. Leave it there. And we also have this discard unmodified counterpart peptides for protein quantification. So this is under the global parameters under protein quantification, and so you can see here it says use only unmodified and for use in protein quantification. And by default, oxidation and protein N-terminal acetylation here are turned on. So we want those peptides to contribute to protein quantification because we don't think that they are going to be differential between the samples. However, we don't want our counterpart peptides for the phosphotyrosines or other PTMs to contribute to the protein quantification. So if I just give you an example, let's just assume we're working with SILAC ratios for a second. So if I have identified 3 peptides that belong to a protein. One of which is a post-translationally modified peptide, one is the counterpart peptide, and one is a third peptide. So if I use both non-modified peptides for protein quantification, it's going to take a median of both of them, and it's going to be incorrect. It's going to be incorrect because that unmodified counterpart peptide has a modified counterpart, right? So it's going to be influenced by the level of the post-translational modification which is on there. Okay. So one last thing that I wanted to say, and so quantification, as I said, is going to be covered in the tutorial. But for now, keep in mind that peptides may be identified with more than a single modification state. So you may have one peptide sequence, so the same amino acid sequence, which is modified on one of the serines, or 2 of the serines, or 3 or more, right? And that is dealt with in, that's output as _1, _2, and _3 in the output table. So that's just something to bear in mind as we go into the tutorial after lunch. And one last thing that I want to mention, which may well be covered later, but when we're analyzing all of our data, all of our PTM data, it's just worth keeping in mind that you need to remember about the protein abundance. So here, I've just plotted increasing protein ratio and increasing post-translational modified ratio. And here we can see, or in this yellow box, we have correlated protein and PTM changes, right? So you think you've found a novel modified site, but actually it's just because the protein has increased to the same extent or decreased to the same extent. So you're much more interested in things that lie outside of that axis. So this purple dot, for example, this has an increased post-translational modification ratio, but a decreased protein. So how does that happen, right? Well, this could be ubiquitin, for example, right? So ubiquitin, your protein has been ubiquitinated to target it for degradation, and some of the protein has been degraded, but the remainder is still sitting around waiting to be degraded. And this one is, for example, a protein which has not changed in abundance, but has become heavily phosphorylated. Okay, so with that, I'd like to thank the Cox Group and the organizers of the summer school for allowing me to be here and enjoy this wonderful place that we have. So thank you very much.
Thank you, Dan. And if you have any questions.
Yeah. I have a question. I don't really, know how the search works in Mars-Quant, but my question is, so when you choose PTM, you choose the STY or ST. So
Yeah.
would there be an advantage in having an option where you just look for Y? Because that's a completely different. So is there any difference, or is that the way?
Yeah,
works?
definitely. So by having all 3 amino acids as kind of potentially being modified,
Right.
You have more search space, right?
Right.
So the reason that it's set up as only serine threonine or serine threonine tyrosine is because in standard enrichments, we don't actually get that many tyrosine phosphorylated peptides. So there you could just reduce the search space and use serine threonine alone. The reverse is not so true. So when we look for just tyrosines, actually you see quite a lot of the peptides also contain a phosphoserine and a phosphothreonine. So if you use just tyrosine, you would probably lose a lot of the tyrosine phosphorylated peptides.
Okay. And the second question I had with reference to abundance versus occupancy. So when you're doing an enrichment, isn't it true that the enrichment reflects either occupancy increase or abundance increase. Is it okay to assume that if you're having a large peptide enrichment for one peptide, it reflects increased abundance or increased occupancy?
Yes, so that's why you have to be very consistent with your enrichment steps.
Hi, thank you. Thank you for the nice talk. My question is regarding UVPD, I mean ultraviolet photodissociation. Is it MaxQuant able to process this kind of data, which is like full of a wide range of ions, and is nowadays being very, very used or increasingly used to study these PTMs as well?
Yeah, in principle, it's possible, but I think the scoring for UVPD still needs to be improved in MaxQuant. So.
Yeah.
This might be a stupid question, but if you've got a modification which you know is going to inhibit trypsin cleaving there, how does MaxQuant deal with that in the fact that the unmodified peptide is going to be a completely different length?
So there's this feature in there for to specify the number of miscleavages which you're prepared to accept. So if you know that your modification, let's say, is on a lysine, and you know that that's going to cause trypsin to miss cleaving after that lysine, then you should probably increase the number of miscleavages which you allow in the search. So by default, it's set as 2, but maybe you could increase it to 3.
Thank you for the nice talk. I have two curiosity. One is, in the deep proteome analysis, you were showing that there are 10,000 sites for phospho and 7,000 for N-acetylation. Would you recommend to always run in a regular search also phospho identification?
If you've done really deep proteomics and you're interested in the quantification of those sites, it's definitely worth turning on, yeah. You could also do a dependent peptide search to see how, maybe on a few, so maybe on one of the replicates, right? So if you have 10 replicates with 8 high pH fractions, maybe you could take one of them and do a dependent peptide search. And by that, you would maybe know which modifications should I turn on in the variable modification search.
And the other question is, if you could explain more why we exclude the quantification based on the unmodified peptide, because in your experiments of the 3 peptides,
yeah.
So if you take the medium of the modified and unmodified, it should stay the same, right? I mean, if one increase and the other, yeah.
Sorry, yeah. So if we have that situation, right, where you have 2 non-modified peptides, one of which is a counterpart and one of which is just another peptide on the protein. If the counterpart peptide has, if the modified peptide that is the counterpart has a ratio on it, right, the strong ratio, then you expect the unmodified counterpart to have a different ratio. So it's going to have gone down or gone up dramatically. And then the third peptide, which doesn't have any modification sites on it, right? Let's say this is 1-to-1, right? The protein hasn't changed. So now it's going to take a median of a ratio of 1 and a ratio of 10. And that's going to be wrong in terms of protein quantification.
Thank you for the talk. I have a question. For example, if we have a phospholipid-enriched sample, and would it be fair to give, if we're sure that we have, for example, 90% phospholipid in there, would it be fair to give a higher score to the phospholipid as compared to the non-phosphorylated peptides, and are there the ways to do that simple, or you wouldn't do this at all?
So you're trying to say limit the search space only to phosphopeptides?
Yes, or yeah, initially give them higher score than the not phosphorylated peptides.
So I don't think you would necessarily get many more phosphopeptide identifications using that strategy. I can't think of a way that it's possible to do that in MaxQuant to turn off standard peptide identification and just keep STY. We have. I mean, you'll see in MaxQuant there's fixed modifications, but that can't be used for phospho-STY because then MaxQuant will expect it to occur on all serine, threonines, and tyrosines.
Okay, thank you.
Sorry.
Hi, thanks for the talk. I have a question regarding the occupancies working with TMT data, for instance, because you can imagine that the ratio compression will be very different in a deep proteome versus the phospho-enriched sample, for instance. How robust would you say is MaxQuant there?
I don't know if MaxQuant, it doesn't do occupancy calculations for TMT, but this paper, this benchmarking paper from Jesper that was just published, this has it has a strategy in there for deriving occupancies from phospho-TMT data. So that's not implemented in MaxQuant. I don't know if it will be, but you could use that to derive your own occupancies in some other software.
Thanks.